Welcome to the Data Analysis Real-Time Quantitative PCR tutorial presented to you by the Bio-rad Life Science Group Technical Support Team. This tutorial covers the basics of analyzing real-time quantitative PCR data. It includes information on both absolute and relative quantification methods. Real-time quantitative PCR can be performed on several different platforms, depending on the instrument you are working with. The methods presented here can be applied to virtually any real-time platform, including both the Bio-rad and MJ systems. In conventional PCR, the amplicon is detected by endpoint analysis. In real-time PCR, the accumulation of the amplicon is detected and measured as the reaction progresses, that is, in real time. This amplification plot shows the fluorescence from the amplification reaction, which is proportional to the amount of amplified product on the y-axis. The PCR cycle number is shown on the x-axis. During the early cycles, fluorescence remains at background levels. Eventually, however, enough amplified product accumulates to generate a detectable signal. The cycle number at which this occurs is the threshold cycle, or CT. In this plot, the threshold fluorescence is indicated by the green line. In real-time quantitative PCR, the data that are generated are the threshold cycle, or CT values. The threshold cycle is determined mainly by the amount of template present at the start of the amplification reaction. If a large amount of template is present at the start of the reaction, relatively few cycles are required to generate enough product to give a signal above background. Thus, the reaction will have a low or early CT. Conversely, small amounts of template generate larger or later CT values. This relationship forms the basis for the quantitative aspect of real-time PCR. In this plot, then, the different curves are generated by a twofold dilution series of starting material. The most dilute samples generated the curves toward the right. When analyzing real-time data, there are two methods to choose from: absolute quantification and relative quantification. The choice of method is based largely on the experimental goals and on available resources. Absolute quantification is used when an experimenter wants to know the properties intrinsic to a given sample. For absolute quantification, the question often asked is, how many? Two examples of applications in which absolute quantification are used are copy number determination of either chromosome or gene copy number and viral load measurement. In both of these examples, the measurements yield a quantity of nucleic acid, for example a copy number or micrograms, per given amount of sample. Absolute quantification is achieved by comparing CT values of test samples to a standard curve. To construct the standard curve, different known amounts of a template are analyzed by real-time PCR. The resulting CT values are plotted on the y-axis against the log of the starting quantity of sample. In this example, these values are represented by the black dots. A linear regression analysis of the data generates an equation, which is then used to derive the quantity of the test samples, samples A and B here. For this approach to be effective, the CT values for the samples must be within the range of the standard curve. The standard curve gives a straight line when we view it with CT plotted against the log of the starting quantity. Y equals mx plus b is simply the equation for a straight line. Here, m is the slope, x is the log quantity, and y is the CT value. From the equation for the linear regression, we can derive the following equation to determine the quantity, n, of an unknown sample, little n. The quantity of copies in sample little n is equal to 10 raised to the power of the quantity little n minus b over m. Little n in this equation is the CT value for the sample little n. Note that the units drop out of the equation. In other words, Whether you choose to use copies, viruses, genomes, moles, grams, or any other unit, the type of unit does not matter as long as you use the same unit for every point on the curve. When looking at different data sets, however, work with the same units for each experiment you plan to compare. This table shows an example of the determination of absolute copy number using absolute quantification. The copy numbers for two samples, A and B, were determined using the measured CT values and the equation derived from a standard curve. Typically, when replicates are sampled, a mean and standard deviation of the individual samples are also reported. Note that the standard curve may only be used to interpolate, not extrapolate, the quantity of the unknown sample. This is because the assay may not be linear outside the range covered by the standards tested. The mathematics of absolute quantification are easy to perform. The method does, however, require a reliable source of a template of known concentration, and standards must be amplified in parallel with the samples every time the experiment is performed. This means that you must use some other method to measure the concentrations of your standards. Absolute quantification answers the question, how many? However, it does not provide direct answers about changes or differences in gene expression. Relative quantification is used to compare changes in gene expression, where the question asked is, what is the fold difference? How much is gene A expressed here rather than there? This type of analysis can compare expression of multiple genes, and it is usually performed with more than one gene. Almost anyone interested in gene expression and regulation is interested in relative quantification methods. Three examples of applications in which relative quantification might play a part are siRNA, or RNAi-induced gene silencing, developmental biology, and disease research. Where absolute quantification required a standard template of known concentration, relative quantification requires a calibrator sample. The calibrator sample ensures that the comparison is made between equivalent amounts of starting sample. Two methods are most commonly used for relative quantification: normalization against a reference unit mass and normalization against a reference gene. The first of these methods is normalization against a reference unit of mass. This method uses a unit mass, such as a cell number or amount of nucleic acid, as the calibrator, and so requires accurate quantification of the starting material. Usually, one sample is chosen as the calibrator or control, and the expression of the target gene in all other samples is expressed as an increase or decrease relative to that calibrator. The calibrator is usually the experimental control. When using this approach, use the same amount of mass for each sample. Carefully count the number of cells or amount of tissue that is used, or carefully measure the starting amount of RNA that is added into the reactions. After PCR, the CT values for the test sample and calibrator are used to calculate the ratio between them using the following equation, where E is the efficiency of the reaction, which is calculated using the slope of a standard curve. If we assume that efficiency is perfect, or 100%, then E in the equation simplifies to 2, and we can rearrange the equation as follows. The ratio is equal to 2 raised to the power of delta CT, where delta CT is the difference in CT values between the calibrator and test sample. This equation can be entered into a spreadsheet for automated analysis of multiple samples. Here is an example examining the relative expression of a gene in a healthy control sample and in a tumor sample. The control yielded a CT of 15, and the tumor a CT of 12. Using these values with the equation we derived on the previous slide, we find that the expression of this gene is 8-fold higher in the tumor cells than in the healthy control cells. This method is simple, but does have some limitations. It requires accurate quantification of the starting material. It assumes knowledge of the reaction efficiency and that the efficiency is approximately 100%. It also assumes that there are few changes in total gene expression between the sample and the control. Some of these limitations can be overcome by using normalization against a reference gene. Normalization against a reference gene circumvents the need for accurate quantification of the starting material. This is advantageous in studies where starting material is limited. The drawback is that the method requires at least one known reference gene that exhibits constant expression levels in all samples and whose expression is not changed by any treatments that are being investigated. The identification of such a gene is not trivial, and some have even proposed that, in most cases, the use of multiple reference genes may be necessary for accurate quantification. As with the unit mass method, at least one calibrator sample is required in addition to the test samples. The method also requires at least one reference gene and at least one target gene. The target gene is also referred to as the gene of interest. To use this method, you must first determine the expression levels of the target and reference genes in all sample types using RT-qPCR. We will demonstrate the calculation for the most simple experimental design, one that uses a single test and a single calibrator sample, and a single target and single reference gene. In this scenario, 4 CT values are determined. The three most common approaches to normalization against a reference gene are the Levac method, also known as the delta-delta CT method, the delta-CT method using a reference gene, and the FOFL method. Each method has its advantages and disadvantages, as well as assumptions that must be met. The Levac method requires that the target and reference genes amplify with high efficiencies, and that the efficiencies differ from each other by less than 5%. Most optimized reactions fall within these criteria. If reaction efficiencies are low, optimize the reactions and, if necessary, select different primer pairs. The Levac method is also called the delta-delta CT or double CT method because it requires the determination of two delta CT or difference in CT values. In the first step, normalize the CT of the target gene to the CT of the reference gene. In the second step, normalize the delta CT of the test sample to the delta CT of the calibrator sample. In the third step, calculate the expression ratio or fold difference. In this example of the Levac method, we examine the CT values for the target gene, P53, and for the reference gene, GAPDH. We examine the expression levels of these genes in a control sample, which is the calibrator, and in a tumor sample, which is the test sample. The CT values for each gene and for each sample are entered into the table. Next, the CT values of the target gene in both sample types is normalized against the CT value for the reference gene. To do this, subtract the CT value for the reference gene from the CT value for the target gene. In this example, we obtain negative values for both sample types. Next, we normalize the delta CT of the test sample to the delta CT of the calibrator. In other words, we subtract the delta CT of the calibrator from the delta CT of the test sample. The value obtained represents the change in expression of the gene of interest between the test and calibrator conditions, normalized for any differences in loading between the reference and test samples. Given this delta-delta CT value, next determine the fold difference in expression between these two genes in the two samples. Use this equation. In this example, the equation yields a value of 5.3, indicating that tumor cells express P53 at a 5.3-fold higher level than the control cells. Negative values indicate a difference in expression that is lower than the control sample. The delta CT method is a variation of Levac method that is simpler to perform but that generates essentially the same results. This method uses calculations for the difference between the reference and target CT values for each sample. It also uses an expression value for the calibrator sample that is not 1.0, which is the main difference between this method and the Levac method. It also generates results identical to those from the Levac method if the results are divided by the expression value of the calibrator. Using the same genes, sample types, and CT values we used to demonstrate the Levac method, we first determine the relative expression of the two genes for each sample using this equation. For the control cells, this yields a value of 2.8, and for tumor cells, a value of 14.9. These values represent the expression of p53 relative to GAP-DH in both samples. At first, these values appear to be very different to those we determined using the Levac method. However, a quick evaluation of the ratio between the two sample types reveals that the two methods generate the same results. The Levac method is useful only when the reaction efficiencies of the target and reference genes are similar. If these efficiencies are not the same, an alternative formula must be used to determine the relative expression of the target gene in different samples. The FOFL method uses the following equation to determine the expression ratio between the test and calibrator samples. E target and E reference are the reaction efficiencies of the target gene and reference gene, respectively. This equation assumes that each gene has the same amplification efficiency in the test and calibrator samples. The FOFL and Levac methods are closely related. In fact, the Levac method is a special case of the FOFL method in which the efficiencies of the genes are both 100%. In conclusion, for RT-qPCR, data analysis requires data generated in optimized specific reactions. Absolute or relative quantification methods can be used. Of the relative quantification methods, the FOFL method is the most powerful, but it simplifies to the Levac method. The Levac method is the most commonly used relative quantification method. Finally, all data analysis methods are based on CT values, making them available for use with any reaction chemistry or platform. We hope that this tutorial answered your questions. For more detailed information about real-time PCR, refer to the Real-Time PCR Applications Guide, which is available for purchase from Bio-rad using the catalog number shown here. For more information and troubleshooting advice, visit the Gene Expression Gateway at biorad.com/genomics. For further inquiries or troubleshooting assistance regarding the content presented in this tutorial, please contact the Bio-rad Life Science Group Technical Support Team at the phone number or email address shown here. Technical support is available in the United States Monday through Friday, 7 o'clock a.m. to 5 o'clock p.m. Pacific Time. For local technical support offices or to access the Frequently Asked Questions database, go to consult.biorad.com. Thank you for taking the time to view this tutorial. Your feedback is important to us. If you would like to share your comments with us, please use the email address shown here.